Good day grade 12 learners, and welcome to this video where we will be looking at the entire process of protein synthesis. So in the previous video we would have touched on RNA location, structure and functions. We looked at the location of RNA, the structure of RNA, three components of an RNA nucleotide, a stick diagram of mRNA and tRNA. And then lastly, we see the function of RNA is that RNA plays a role in protein synthesis. In today's video, based on the exam guidelines, we will look at the entire process of protein synthesis. We will look at the involvement of DNA and RNA in protein synthesis. We will notice that protein synthesis involves two major stages. That is stage number 1, transcription. The second one being translation. So we will describe the entire process of transcription. We will describe the entire process of translation. And we will then look at a simple diagram to illustrate transcription and translation in protein synthesis. So we just have a look at the overview of protein synthesis. Like I've mentioned a few minutes ago, transcription is going to be the first phase of protein synthesis, and transcription takes place inside the nucleus. If we just have to give a general overview as to what happens in transcription, we can say that DNA serves as a template to assemble a single strand of mRNA from the cell's pool of free nucleotides. Afterwards, RNA moves into the cytoplasm. So what exactly does this mean? We are using the DNA as a template strand to assemble a strand of mRNA. And to do that, we are going to use the free-floating RNA nucleotides from the nucleoplasm. Thereafter, the mRNA strand is going to move to the cytoplasm and it's going to attach itself to the ribosome. The next phase of protein synthesis will be translation. Translation occurs at the cytoplasm, specifically at the ribosome. What happens in translation? mRNA and tRNA is going to control the assembly of amino acids into proteins. So in summary, if we have to look at what we just discussed, we started with DNA. We moved over to the process of transcription where we formed our mRNA strand. We then underwent translation to form the protein, and we want to end off with forming a protein as we are looking at protein synthesis. So this is the diagram that we will be using to describe the entire process of protein synthesis. We are first going to describe transcription and then we will describe translation. So if we are going to firstly describe transcription, now before I start with this, just something that you need to take note of. You will be asked to describe the process of transcription or the process of translation in a test or in an exam. This question could be for about 4 to 6 marks. And in your attempt to answer this question, please make sure that all the information that you are stating for your process of transcription and translation, you take it exactly from the exam guidelines. So in the exam guidelines that I initially had, the second slide of this video that had the exam guidelines that describe protein synthesis, you study transcription and translation from the exam guidelines. The reason as to why I'm saying so is because the memos to the papers are generally constructed from the exam guidelines and specifically where there are processes. If the process is described in the exam guidelines, then you study it from the exam guidelines. I know that you'll have access to a lot of other notes, like notes that your educators are issuing to you in class. You'll also have access to notes from textbooks. However, if you want full marks for answering a question on transcription or translation, please study it straight from the exam guidelines. So firstly, transcription takes place inside the nucleus. Now, why does this process start inside the nucleus? You'd have remembered in the previous video when we looked at the process of DNA replication. We said that DNA replication took place where? Inside the nucleus. And in which phase of a cell cycle did it occur? In interphase of a cell cycle. So since the process of DNA replication occurred inside this nucleus, it therefore follows that that next process that we are looking at now to start protein synthesis, transcription, it will also occur inside the nucleus. So step number one, if you are describing transcription, you're first going to start with your DNA strand, your DNA double helix structure that you had at the end of DNA replication. That DNA double helix, it's going to start unwinding and unzipping. So the DNA double helix strand, it's going to start unwinding and unzipping. The weak hydrogen bonds that are generally formed between these nitrogenous bases, that weak hydrogen bonds are going to break. And when the weak hydrogen bonds break, your DNA is going to form two separate strands. So that DNA strand has now formed two separate strands. I'm going to go again from the start. You had a DNA double helix structure. The DNA double helix unwinds and unzips, and the weak hydrogen bonds between the nitrogenous bases are breaking, so that you have two separate strands forming. Only one of these strands are going to serve as a template strand, and it serves as a template strand to form mRNA. You are then going to have free-floating nucleotides coming from the nucleoplasm here, because we're inside the nucleus. Free-floating nucleotides coming from the nucleoplasm. They're going to align themselves complementary to this DNA template strand, so that they form an mRNA strand. Once they have formed an mRNA strand, then this is what the mRNA strand will look like. The mRNA strand will now leave the nucleus through the nuclear pore. It will go to the cytoplasm to start the next process, which is translation. If you go down here on the left, you will see I've described the entire process of transcription. The DNA double helix unwinds and unzips. The weak hydrogen bonds break. You're going to have two separate strands forming, but only one DNA strand serves as a template to form mRNA. You're going to use free RNA nucleotides from the nucleoplasm. The mRNA will be complementary to the DNA strand. And the mRNA strand now has the coded message for protein synthesis. What does that mean? The single mRNA strand that you have formed during transcription, it's going to comprise of nitrogenous bases that you would normally have on an RNA molecule. Therefore, you have uracil. You don't see thymine on the strand. Where would you have seen thymine? You would only have seen it on the DNA strand. Now, if we just take a group of 3 of these nitrogenous bases found on the mRNA strand, then this group of 3 nitrogenous bases, namely uracil, cytosine, adenine, it's referred to as a codon, and this is something that you need to know. So that's the first codon, second codon, third codon. A codon is a group of 3 nitrogenous bases found on the mRNA strand. And these codons is what's going to provide the coded message to start protein synthesis, so that we can move over to the next process, which is translation. If you take a look at this following video, it can assist in your understanding of the process of transcription. So originally you can see you have your DNA strand here, after transcription of the mRNA is formed, it's going to coil up. mRNA strand is coming, it's still in the nucleus, it wants to leave the nucleus to the nuclear pore and go attach itself there to the ribosome to start translation. So if we move on to the next part of protein synthesis, translation. Translation occurs in the cytoplasm. And it's generally going to occur specifically at the ribosome. Another important thing that you need to remember is the ribosome is known as the site of protein synthesis. The ribosome is known as the site of protein synthesis. I'm going to first use this diagram to explain translation to you. So we came out of the nucleus. Remember that mRNA formed inside the nucleus, left the nucleus through the nuclear pore and went to the cytoplasm to attach itself to the ribosome. Here you can see the mRNA strand has attached itself to this ribosome. The mRNA strand is attached to the ribosome. You can see it has that group of 3 nitrogenous bases, UCA, forming the codon, another codon, another codon. We can now start the next process, which is translation. So what happens in translation? Firstly, you are going to have a tRNA strand. You're going to have a tRNA molecule coming. That tRNA strand, it's going to carry 2 things. Firstly, it's going to carry an anticodon. It's going to carry an anticodon. Anticodon is opposite of codon. So remember on the mRNA strand, you had a codon. So now the tRNA strand must bring an anticodon. So what would the anticodon possibly be? It's going to be a group of 3 nitrogenous bases found on the tRNA strand that's going to be complementary to the codon on the mRNA strand. So if we have a look at the mRNA strand, the first codon, we have UCA. The complementary anticodon, which is going to be complementary to that, should be AGU. So if on the mRNA strand we have a codon UCA, then the anticodon, which is complementary to that, will be AGU. So what exactly happens in translation? Your tRNA strand is going to bring an anticodon and an amino acid. Now, why is it important for it to actually bring the amino acid? Now, remember earlier on when we spoke about the building blocks of DNA, we said the building blocks of DNA is going to be the nucleotide. Now, the building block of a protein is going to be an amino acid. And because we are trying to make a protein here in the process of protein synthesis, it therefore follows that we need to have amino acids somewhere along this process. So we did not have amino acids in the process of transcription, but now in translation, that is where we are bringing the amino acids. So the tRNA is going to carry an anticodon and an amino acid. When the tRNA strand brings the anticodon and aligns it complementary to the codon on the mRNA strand, it also brings an amino acid. All these amino acids are eventually going to join together using a peptide bond. This is then going to form a polypeptide chain which forms the required protein that is formed. And the process of protein synthesis is now complete. I'm going to take you through it here on the left hand side. Each tRNA carries a specific amino acid. When the anticodon on the tRNA matches the codon on the mRNA, then the tRNA brings the required amino acid to the ribosome. Amino acids become attached by peptide bonds to form the required protein. So the tRNA is bringing the anticodon complementary to the codon on the mRNA. The amino acid gets brought to the ribosome. Amino acids get joined by peptide bond to form the required protein that is formed. So that is the process of translation. So if you have a look over here, you can see that this is a tRNA strand carrying all its anticodons. Here you have an anticodon UAC. It's matching the codon on the mRNA strand AUG. And at the end, it also carries an amino acid That's the name of the amino acid here. All the amino acids will join to each other to form the required protein. They will join using a peptide bond. So if you have a look at the following diagram, it can show you the entire process of protein synthesis. Firstly, inside the nucleus, you have transcription, and then in the cytoplasm at the ribosome, you are going to have translation. So these were the two stages of protein synthesis that we touched on, transcription and translation. So in summary of today's video, based on the exam guidelines, we looked at the process of protein synthesis, the involvement of DNA and RNA in protein synthesis. We said transcription is where the DNA double helix unwinds and unzips. The weak hydrogen bonds break to form two separate strands. One strand is used as a template to form mRNA using free RNA nucleotides from the nucleoplasm. The mRNA is complementary to DNA. mRNA now is the coded message for protein synthesis. So mRNA moves from the nucleus to the cytoplasm, attaches itself to the ribosome. Next process, translation. Each tRNA carries a specific amino acid. When the anticodon on the tRNA matches the codon on mRNA, then tRNA brings the required amino acid to the ribosome. The names of the codons, anticodons, and amino acids are not to be memorized, so please don't memorize any names of codons, anticodons, or amino acids. The names of the amino acids will always be given to you in the exam. Amino acids become attached by each other by peptide bonds to form the required protein. And then we look at the simple diagram to illustrate transcription and translation. Just a reminder, please make sure you are studying transcription and translation from the exam guidelines. These are easy marks that you can have in your bag for paper 2 of your final exam. So that concludes the entire topic of DNA, the code of life. We have touched on various subsections in DNA, the code of life, the last one being protein synthesis. In the next video, we'll move on to the next topic that is going to be a topic covered in paper 2. That video will start the topic on meiosis.